Have you ever heard of an oak apple gall?
I have not.
If you were to guess, what would you think it is?
Well, my first instinct is like a tumor on a tree, like an oak tree, or something to do with the gallbladder.
Clearly, the public is lacking knowledge about the oak apple gall. Let's take a closer look.
Oak apple gall is a disease commonly afflicting black, red, and scarlet oaks, where spherical protrusions are formed by members of the cinnamid wasp family Cynnipidae during reproduction. These golf ball-sized galls are often compared to apples hanging from a tree, hence their eponymous namesake. To better understand these oak apple galls, let's first explore their developmental process. Oak apple galls have 3 stages of development: initiation, growth, and maturation. The initiation phase, for starters, begins with the oviposition of a mother wasp's eggs within oak meristematic tissue. Stem cells in the meristematic tissue, coupled with egg secretions, are utilized to initiate gall growth. The oak host responds to this gall induction by lysing cells underneath the eggs. This plant cell necrosis leads to rapid cell proliferation and differentiation of the cells inferior to the lysed site, creating multi-layered chambers for larvae maturation. An example chamber is shown in the figure on the right. Once hatched, larvae enter their individual chambers to control further tissue differentiation.
As the larvae mature, the growth phase is initiated to keep up with their metabolic demands via cell expansion of the chambers. The larvae start to feed on the inner gall tissue, consisting primarily of vacuolate parenchyma cells. These cells contain high levels of starch and low levels of lipids to provide initial nutrients to growing larvae. Larval presence induces endoreduplication, or polyploidization, of the nuclei, producing multiple copies of the chromosomes. The nucleus now has rapid rates of transcription and translation, increasing photosynthetic machinery and protein production for the growing larva. Products from greater photosynthetic rates in neighboring oak leaves are intercepted and routed to the larva. The plant also produces proteins that signal the invasion of the foreign larval invader. One protein that is released by the plant during times of stress is called NAD+ dependent formate dehydrogenase, also known as FDH. FDH is an enzyme that catalyzes the oxidation of formate ions to carbon dioxide, as depicted in this diagram. The oxidation reaction is coupled with the reduction of NAD+ to NADH. NADH is an electron donor in the electron transport chain, which makes ATP for the cell to use under the stressful circumstance of larval invasion. Although gall formation signals a stress response in the plant, the gall itself does not pose as a threat to the health of the plant. As the larvae continue to feed on the vacuolate parenchyma cells and increase in size, the inner gall tissue is depleted and can no longer sustain the larva. This initiates the transformation of vacuolate parenchyma cells into inner nutritive cells. These cells are also incredibly high in nutrients, containing high levels of lipids and proteins and low levels of starch. A major inducer of increased lipid content is the biotin carboxyl carrier protein, also known as BCCP. An increase in BCCP production by the plant is controlled by the maturing larva. This protein is a subunit of the class II acetyl-CoA carboxylase, also known as ACCase. It catalyzes the carboxylation of acetyl-CoA into malanyl-CoA via an ATP-coupled reaction. BCCP is covalently bound to lysine, which acts as an arm to transport acetyl-CoA to different active sites of ACCase. Malanyl-CoA plays a key role in chain elongation during fatty acid biosynthesis. The increase in larva-induced lipid production by BCCP provides enough nutrients for the larva to enter into the next phase of development, maturation.
During this stage, cell division rates slow down and the chamber stops receiving major influxes of host nutrients. The larvae continue to develop and feed through nutritive cells until the sclerenchyma cell layer surrounding the vacuolate parenchyma is reached. While these internal events are occurring, tissue surrounding the outer gall lignifies and becomes tough and woody in texture. This lignification discourages predation by herbivores and, in most cases, acts as a temporal marker preceding gall shedding from the oak host during the pre-autumn season. Once the gall drops to the ground, desiccation of the structure itself follows, and subsequent leaf coverage becomes the next adequate microclimate for the larvae to either pupate and continue development into adult wasps, or undergo a period of suspended growth called diapause if environmental conditions are unfavorable.
Let's now take a look at the biological significance of oak apple galls. Because the majority of oak apple galls are usually only the size of a golf ball, the formation of apple galls does not significantly harm oak trees. The apple galls are so small in comparison to the massive oaks that even extreme infestation causes only minor effects, with heavily infested trees often dropping their leaves slightly earlier than normal as the most notable effect. This could be due to the gall concentrating energy from the localized area, leaving less resources for the leaves, resulting in premature leaf fall. But while the galls may be insignificant to the oak tree, apple galls are absolutely crucial for gall wasps and a whole community of insects. Galls can be formed by a number of wasps in the Cynipidae family. Over 1,000 species of cynipid wasps are known to form apple galls, each creating a distinct gall that varies in shape and size. Gall wasps form apple galls during their sexual stage of reproduction. Let's take a look at the life cycle of an example species, Biorhiza pallidae, which alternates between an asexual parthenogenic generation in the summer and sexual generation in the spring. In the spring, females lay eggs on the roots of oak trees. Galls develop on the roots as female larvae hatch and wingless, parthenogenic females develop underground. Once mature in the summer, the females crawl up the oak tree and lay unfertilized eggs in the developing leaf buds. It is here where the oak apple gall forms, and the sexual generation containing males and females develop and emerge in the spring. Sexual females must mate in order to produce the agamic female offspring in the tree roots, once more restarting the cycle. So clearly, oak trees and the apple and root galls they produce are imperative for the survival of cynipid wasps like B. pallida. However, it's not just these little wasps that use the gall's nutritive tissue to support larval development and maturation. Apple galls are actually tiny little ecosystems that often support over 20 different species of insects. All apple galls contain 3 trophic levels: the gall tissues of the plant, gall wasp and inquilins that share their homes, and parasitoid predators. In Latin, the word inquilin means lodger or tenant. So can you guess what inquilin insects are? Inquilins refer to organisms that live commensally in the gall homes of wasps, eating their nutritive tissues of the gall and sharing the shelter without harming the developing wasp. Common inquilins of apple galls include midges, moths, beetles, and other cynipids that have lost the ability to form their own gall homes. Parasitoids are chalcid wasps that attack oak apple wasp larvae and the inquilins harbored within the gall. While a 3-level trophic system is always present, some apple galls even have hyperparasitoids and superparasitoids in a 5-level trophic system, where larger insects or even birds serve as higher-order predators, topping off the complex food web sustained by apple galls. The size of the apple galls determines the parasitoid community, with insects dominating smaller galls and woodpeckers frequenting the larger galls. In some complex communities, mutualistic ants join the party and serve as gall wasp bodyguards that tend to the galls and reduce parasitoid abundance in return for gall-secreted honeydew that attracts the ants. The community dynamic supported by the gall seems almost endless, as each cynipid hosts its own complex ecosystem with varying insect species. Thus, apple galls have served as an important model in ecology. Since all galls consist of at least 3-level trophic systems, apple galls have been used as an important model for trophic cascades, host specificity, and host-parasite interactions in the field of community ecology. As the co-evolution of gall wasps and oak trees dates back 90 million years through the fossil record, the close co-speciation has resulted in an obligate commensalism where gall wasps and the communities they now host cannot exist without the mighty trees. So what is the biological significance of an apple gall? It's a home, a community. It's food, shelter, and a refuge from enemies. It's a necessity for reproduction, and it's a favorite locale for the insect squatters that inhabit it. A harmless infection to some, it's an entire universe to others. The oak apple gall is not simply a disease. An oak apple gall is a miniature world teeming with life.